Hello, welcome to another edition of 5-Minute FRQ, where I take you through an old FRQ on the AP Bio exam in 5 minutes or less. As always, this information is freely available on the College Board's AP Bio website, so if you have any questions, you can look it up there. All right, this is question 5 on the 2013 AP exam. The table below shows the amino acid sequence of the carboxyl terminal segment of a conserved polypeptide from 4 different but related species. Each amino acid is represented by a 3-letter abbreviation, and the amino acid residues in the polypeptide chains are numbered from the amino end to the carboxyl end. Empty cells indicate no amino acid is present. So I'm given the table that shows me 4 different species and shows me 10 different positions where the amino acids are labeled here. There are several terms in the prompt that are not really relevant to the question. The fact that it's the carboxyl terminal segment doesn't really matter. The fact that you're going from the amino end to the carboxyl end doesn't really matter. The gist here is I'm given 4 different species and I'm given the amino acid sequences, and it wants me to know how you go from one species to another. Now, the fact that these are conserved polypeptides, that basically means that the proteins have the same common ancestor, right? That it's basically the same protein in each species, they just have different versions. When I give this question to my kids, I'll have students who misread this, who think that species 1 turned into 2, 2 to 3, and 3 to 4. That's not accurate. The question is saying that species 1 had a branch that became 2, then there was another branch from 1 to 3, and then 1 to 4, okay? So part A, assuming that species 1 is the ancestral species of the group, that's where it says that, explain the most likely genetic change that produced the polypeptide in species 2, and the most likely change to produce the polypeptide in species 3. So species 2, compare 1 and 2. 1 and 2 has a change in position 4. A valine becomes a lysine. Comparing 1 to 3, the change is after position 8, where in species 3, there are no amino acids in 9 and 10, which tells me the protein ended after after position 8. Looking at the rubric, look what they accepted. So for the first point, how do I go from species 1 to 2? So 1 position, the valine became a lysine. All you had to say was that there was a mutation that changed the amino acid at position 4. Now, you probably can guess that it was a substitution or a point mutation. It's a missense mutation. All those are correct, but all you had to say was that it was a mutation that changed position 4. For species 3, the protein ends early. In my head, I immediately think that's a premature stop codon. All you had to say, though, is that there was a mutation that ended the amino acid prematurely after position 8. Now, that is a nonsense mutation. That is a premature stop codon. All those are true, but all you had to say was a mutation that terminated the polypeptide after the valine at position 8. All right. Part B, predict the effects of the mutation on the structure and function of the resulting protein in species 4 and justify your prediction. So species 4, compare 1 to 4. At position 5 and on, it's completely different. All right. So I harp on my kids with this. When you mutate a gene and ask what the protein, or ask how the protein is different, more than likely the protein's going to fold incorrectly. I tell them that the key word there is fold. All right. If you change the structure of the protein, it's going to have a different 3D shape. It's going to fold differently, especially if you go from like non-polar to polar amino acids. And if you change the structure, you're going to change the function. Now, that's what I tell my kids to say. Now, is it possible you have a mutation that doesn't change the structure? Absolutely, that's possible. Could you have a mutation that changes the structure, but it doesn't change the function? That's also possible. So if you look at the rubric, they accepted basically 3 different combinations. Either it has a different structure and a different function, which is what I would say, and the justification is, because you change the structure, it folds differently, which changes the function. You could say that it has a different structure, but it doesn't change the function, and the justification is you change the structure, but it didn't affect the overall function. Or you could say that the structure and function are both not affected because it didn't affect the shape, therefore it doesn't affect the function. So really, any of those work as long as you explain it correctly. But my advice again is to say that if you change the structure, because the protein folds differently, that the function is also different, as in the enzyme is likely non-functional. All right, hope that was helpful, and I'll see you guys next time.